All right, morphology and anatomy of plants. As I was saying, we won't get through all the slides that you have, but that's okay because it may take 3 to do it. All right, now, for starters, plant anatomy, if you Googled it, you'd get something called phytotomy, probably, which I'd never use that word. Plant anatomy is a general term for the study of the internal structure of plants. While originally plant anatomy included plant morphology, which is a description, as you see here, of the physical form and external structure of plants, since the mid-20th century, around the 1950s or so, the investigation of plant anatomy and plant morphology are considered distinctly separate, okay? In short, now plant anatomy is just the internal plant structures, and that's what, but it's frequently looked at, plant anatomy as it's looked at the cellular level, and often involves a sectioning of tissues and looking through the microscope at these slides that you make. Now, back when I took plant anatomy, and I taught it later on, but we used a razor blade. And something we'll talk about called pith, we use pith sticks, which were elderberry pith, and we would put a section of the leaf or stem we were cutting between 2 pith sticks and slice it with a fresh razor blade. After 3 or 4 swipes, a razor blade's not much good because you don't get good thin sections, which brought me to think, remembering a piece of equipment called a microatom, M-I-C-R-O-T-O-M-E. I'm not sure Donna ever had to deal with one of those, but a microatom.
Yes, I have.
Okay. Well, a microatom, I had one in my office when I left Georgia Southern. I should have took it with me because I'm sure they threw it out because they didn't know what the hell it was.
Oh, man.
But it probably cost $300 in its day. But if you bought a microatom today, it would be around $4,000. I looked last night. What it is a fine piece of equipment for cutting ultra thin sections of tissue to look at under the microscope when you mount them on a slide. And there used to be a course, probably 1 or 2 places in the United States today would probably teach a course in histology, H-I-S-T-O-L-O-G-Y. Bottom line is that we're dealing with a lost art of science. In fact, a lot of these supply companies would supply these slides, which are still probably in drawers at a lot of college and universities. Nobody's looking at them anymore, but they're basically the kind of cross sections and longitudinal sections that we'll be looking at today and next time. But I'm gonna share this with you here. Well, I thought it was. Okay, here it is. That is what's the importance of plant anatomy. And I found that on Wikipedia, and I didn't know who the father of plant anatomy was. I honestly didn't know my group. But this is a really interesting point. It's located as a discipline or a discourse. It's located between the study of plant morphology and cell biology. And it allows a student, and I underline these things, they weren't underlined, to conceptually integrate organismal structure and function. And when you think of, well, let me read on. Further, it helps to reveal the relationships between structure, function, taxonomy, ecology, and developmental genetics. Now, when you say function, really you're talking about the way things work, and that means physiology, like plant physiology. So when you talk about structure and function, structure, you're thinking about morphology, and when you're thinking about function, you're thinking about physiology. And my favorite thing to do is identify and classify plants, or reclassify, which means I test what's already been classified to see if I believe it or not. But when we do things like look at the pith, if you look at the cross, most pith is round as we'll talk about, but sometimes in the case of oaks and poplars, it can be star-shaped. Bottom line is anatomy can help you identify plants. But the average, even the average botanist hasn't had anatomy anymore. And most of our botanists in this state haven't had that luxury. And therefore they don't have it. I remember when we were up on a field trip with Ellen Honeycutt, and Ellen's a computer type, but she's a self-taught botanist. But when I remember the look on their faces when I showed them what a chambered pith looks like in walnut with hickory stone. So, but anyway, all right, now let me close out of this useful thing here. Now, all right, any questions so far? All right, we're going to move on through here, and the bottom, I've underlined this. It's not on your slide to remind me to tell you that plant morphology and plant anatomy are used interchangeably. Now, this next slide, the A relates to the B and C on this following slide, and that's why that's coming off of slide 4, which is slide 4 and 5. I could refer back to from time to time, are really the combination 2 slides of everything that's on the 1-page outline I sent you. And that's at Roman numeral 5. There's A, B, and C. And this B and C being together like this implies what I just said, that morphology and anatomy are interchangeable, and that's the way I intend them to be used today and next time. But I found that this is very useful to go back and refer to the biological levels of organization, because this is fundamental to biology. Well, most of you probably had this part of it, this part of biology, that cells make up tissues, tissues make up organs, organs make up organ systems make up organisms, and a lot of places stop there, but you could go on. Organisms make up populations, which are interbreeding groups. Populations are part of communities. Communities are part of the biotic, the living part of ecosystems. Of course, the abiotic parts, the air, etc. And then the ecosystems make up the biosphere, which is a part of the earth where life exists, which is most of it. Now, going back, before I go into this tissues make up organ bit, cells make up tissue. All right. Well, let me go ahead and do it now. The 4 tissues we're going to talk about repeatedly today and next time are meristematic tissues, epidermal tissues, vascular tissues, and ground tissue. Now, you can think of ground tissue as the everything else tissue, because the kitchen sink, you know, what's not epidermis and what's not a vascular is ground tissue. Now, when we get down to organ systems, plants have a root and a shoot system, but they have 3 organs. Actually, they have 3 vegetative organs: root, stem, and leaf. The leaf and the stem are part of the stem system, and the root system is separate. All right, any questions? Now, all right, this first line up here underneath that's not red, cells are the basic structure and physiologic units of plants. That comes from the cell theory concept. The cell theory is all living, majority part of the cell theory is all living things are comprised of cells. Now, of course, viruses would be an exception. There's always an exception. And I'm going to digress for a moment to a tangent that most of you may not like, but it's important in today, and it's because science is in such a disarray, and all this conspiracy theory mess going around. I never in my life would think that people would believe some of the crap they're believing, but here's the relevance. When we say theory, like the cell theory, in the general public, people think that's, when you say theory to the general public, they think it's doesn't have much credibility. You've heard them say, "Oh, that's just a theory," which means that nobody believes it, hardly. But in science, the cell theory is a very highly credible idea, because when you do an hypothesis many times over, usually by different people over many years, it becomes a theory, and eventually a theory becomes a law or a principle. In terms of credibility, acceptability, believability, this is the way it goes in real science. So bottom line is, a theory to a scientist has a high degree of credibility. It's been tested over, a theory represents hypotheses that have been tested for a long time and have a great deal of acceptability. Here's the fly in the ointment. The cell theory is not a theory, it's a principle or law, but the term theory is still stuck. So anyway, the cell theory is really, credibility-wise, the level of a principle or law, but the handle cell theory is stuck for inevitably, we'll be calling it that. Now, all right, any questions about that tangent? It's an important tangent today because people are ignorant about science and technology and don't know the difference. Now, this bullet here with totipotent involves a matching exercise. Now, I'm not going to ask how many did it, but I can guarantee you had problems with it. And part of it is if you don't have those references to look at that I talked about, like the Botany Dictionary. But anyway, we can talk more about that after if you want to try to get them and you hadn't got them. Some of you have tried to get those things. But all right, the answer to totipotent is in the parenthesis, the ability of an organism to come about from one cell, and that's a totipotent, all-powerful cell. The parenchyma cell is the best example of a totipotent cell because it's the one, the parent. Look at the word parent. And chyma is a term that basically means in biology, it's the living tissue of a primary body, in this case, a plant body. So the parent cell from which all other cells are derived is a parenchyma cell. And we're going to use a term many times today, a meristematic cell or a meristem. And parenchyma cells are meristematic. Meristem, and think of stem cells in animals because that's what meristematic cells are analogous to. A meristem is a kind of tissue that has rapid mitotic cell divisions going on. Generally, it's localized in the plant body in the stem, which is the shoot, and root, apical meristems, the apices near the ends. And it constitutes a permanent or temporary zone of actively dividing cells. And then after those cells divide, they mature and differentiate after they elongate. Okay, so it's divide, elongate, and mature, pretty much in that order. We'll point that out again as we go through this presentation. Now, tissues are large organized groups of cells that work together to form a specific function, that is like xylem and phloem and meristematic tissues. Meristematic tissues' function is to divide and form other tissues. Xylem, you probably know, is the water and mineral conducting tissue in plants, and phloem is the food conducting tissue in plants. We'll talk more about those as we go through. And then an organ is a structural and functional unit of a plant, and we've got the root, stem, and leaf, but the caveat is that there's 3 organs, but they're in 2 organ systems because the leaves come from the stem system. And we'll see that as we go through. Now, years ago when I first had this, put this thing out, I didn't like it that much, but the more I think about it, the more I like it. And actually, I sent it as a separate handout because it was so small you couldn't read it. Let me get some water. I've finally gotten through with my seasonal asthma, but I'm still got a little bit of that I suffered with from October to November to my birthday, which is next week, but usually February. All right, but this is busy. It's busy, and but it's useful. It's got some problems. It does show the interconnection between the topics that we said can be used interchangeably, anatomy and morphology, and but the first thing that sticks out to me is right here, and now that's not on your it's not on your slide. It's not on what I sent you last night or night before, I guess. I sent you a one-page deal of this, and I highlighted it here because mesophyll, if you want to see mesophyll, you got to be looking internally in the anatomy of the leaf. And we'll revisit mesophyll once, maybe twice before the day is out to let you understand. but there's primarily going to be, and you don't need to write it down now, but there's going to be palisade mesophyll and spongy mesophyll, which are fundamentally parenchyma tissue. And so notice that this down here is coming off of anatomy. So I'm trying to tie these together. That's what I'm trying to do. I would not have put mesophyll if I had put this concept. It's not my map. I'd have put it over here on this other side. I believe I would. But now, at the bottom left, this is about associating primary growth here in herbaceous plants and secondary growth, which we see in perennials and woody plants. And we'll end up today trying to transition, I think, between primary growth and secondary growth. Most of what we talk about today will be primary growth, and that's growth coming from the apical meristem of the shoot, which is a stem, and the apical meristem of the root. Xylem and Flow, we'll deal with on the next slide in a little more detail. Actually, the next slide we'll actually, we'll concentrate on comparing the differences in the simple tissues and the complex tissues on the next slide. But yeah, that's what that's about. But before we go there, let me say now that the pericycle, if you try to find pericycle, it looks just rather nondescript. And the way to find it is, and it's usually in the roots, not the stems, that you'd find a pericycle, almost unique to roots, is there's a region that we call the endodermis. And that has a lot to do with the slide I've added that we'll get to next time, if not today. But the pericycle is more or less, it's the tissue that forms lateral branch roots. To try to find it out, to recognize it though, you'd never recognize it without this endodermis being next to it, because it looks like nondescript parenchyma. All right, so more on that later. Now, what we're doing now is comparing simple tissues and complex tissues. On the left, it's simple. On the right, the red blocks are complex tissues. Let me get this, I can't make it smaller, but anyhow. All right, first of all, the fundamental cell from which all other cells are derived is called the parenchyma cell. And CH is K, like chameleon. You pronounce it like chameleon. There's no church in biology. Parent fluid is really, that's what this word really literally means. But the cell is more or less isodiametric, which means it has dimensions that are length to width wise about the same, and the third dimension too, X, Y, and Z, about the same. And it can function in starch, or it can be a secretory cell, but usually the parenchyma cell's claim to fame is that it divides and is meristematic, and it's totipotent. Now, it's usually thin walls. Sometimes they can live in cactus. Some cactuses, this is far out to do it, but parenchyma cells can be 100 years old. I don't know how they were able to determine that, but I know some of the saguaros can live to 200 or 300. But anyway, think of soap bubbles, and I don't mean the top of the soap bubble in a bucket. Think of the bubbles down underneath, and that's kind of what you have, this kind of shape. Now, collenchyma, and you'll probably see this view of it on your slide, because that shows the more characteristic look. Because people think of collenchyma sticking corners, but truth is, collenchyma tissue is made up of cells that were parenchyma that have elongated and have uneven thickenings in the walls of the primary wall. Now, primary walls are something that animals don't have walls, cell walls. They have cell membranes. Well, plants have cell walls, and if the walls become, the primary wall also has something following it called a secondary wall, we start talking about sclerenchyma. Now, sclere means hard, just like in the eye. And sclerenchyma tissue is not only primary, but secondary wall thickenings, is what you see here, okay? and they have a tiny lumen, a cavity, tinier than the parenchyma. So primary walls thickened unevenly, and here we have secondary walls developed, which weren't in these other two. Once you reach this sclerenchyma stage of differentiation or maturation or development, your cell is no longer totipotent. It can't de-differentiate and form something else. In other words, once you get to a sclerenchyma stage of development, you're done in terms of forming something else. Now, before we leave this simple tissue thing, we're going to move down here to talk about sclerids or stone cells. Now, there's two major forms that sclerenchyma takes. It takes the form of a fiber, which is like this, but has an ending to it that looks like that, very pointed. This is xylem fiber. But a sclerenchyma fiber, it's not in the xylem, obviously. It has a point on it like that, but in cross-section it has a very thick wall and a tiny little cavity or lumen, like you see here. Think 3D. And then when we compare that to a stone cell or sclerid, there's the only thing they have in common is the tiny little lumen, tiny little cavity. Now, these sclerids or stone cells are kind of like the parenchyma that they're situated with. They are isodiametric. And if you want to think what one really looks like, bite into a pear. Because pears have sclerids or stone cells, and that's what this is from. This is stone pear tissue that we would be looking at under a microscope slide. You bit into an apple, it may crunch, but not the same way. Apples don't have stone cells, pears do. Pears and apples are really closely related. Different genera, Pyrus and Malus, pear and apple. But anyway, now, any questions before we talk about complex tissues? Okay. Now, complex tissues, by definition, well, simple tissues are composed of only one kind of cell. Parenchyma tissue is made up of parenchyma cells. And we can say the same about these other two, collenchyma and sclerenchyma. However, complex tissues are made up of, by definition, more than one kind of cell. And the fundamental cell of xylem and phloem is xylem parenchyma, which is what this is. It's not labeled, but if you got some way to label that, well, actually it is, the label is there. I just can't see it because of the way this screen is doing, but xylem parenchyma is that cell there. Now, it doesn't say so up here, but this is what has come from a phloem parenchyma cell. Now, the counterpart to this phloem cell, which is called a sieve tube element. The counterpart down here is a vessel element. That's what this is. Vessel element corresponds to a sieve tube element. Now, there is in the xylem, all right, in other words, what you have to realize is this vessel element came from a parenchyma cell that was differentiated. Another form of differentiation is over here into a trachid. Now, this is a Tracheid. This is a trachid. And it has little cavities or holes in it, pits. The pits is what these are circular holes. And in gymnosperms, by the way, gymnosperms have tracheads, but they don't have vessel elements. It's like comparing a garden hose to a conduit. Angiosperms have both, and the angiosperms are more efficient conductors of water and minerals because the water moves straight through this vessel element. If you put a bunch of vessel elements together, you've got a structure that you can see with your naked eye, barely. It's called a vessel. So vessels are comprised of what? Vessel elements. Just like sieve tubes in the phloem are composed of sieve tube elements, okay? At the end of a sieve tube element is called a sieve plate. And it's like a shower drain up here. It's like, and that's how stuff moves through the phloem. The stuff is usually sucrose, a disaccharide carbohydrate. That's the way it moves in plants, it being food. Sucrose is translocatable in the phloem. What controls it is this companion cell that you see here. Controls the movement of things. This is important, what I'm about to say. Phloem functions in the living form, and this companion cell controls, like in the sap rising in the spring, it would come up. And then when the storage of that food that's made in the leaves and stored in the roots is translocated down in the fall. So it can move both ways. Frankly, it actually can move both ways at the same time. By the way, this is a cross-sectional view. Companion cell, sieve tube element, cross-section right here. Think 3D. In fact, all three of these, think companion cell, companion cell, companion cell, sieve tube element, sieve tube element, sieve tube element. Now down here in the xylem, xylem can function, which we'll later learn today, as wood. Xylem can function as a dead, when it's dead, and water and minerals move up even in dead cells and without any companion cell control. There's no need for it. And that gets into physiology of something called translational pull. I've got a, boy, it's already 10:30. I can't believe it. Anyhow, all right. So that's a lot of stuff. Have I confused anybody? Because this would be a good time for a question if you've got one. Okay, because that's some heavy duty talking I just did. Because I told you a lot of what goes on functionally in the phloem and xylem and why it's complex, and whereas this stuff over here is simple. Now, the one thing I haven't mentioned is that we talked about sclerenchyma fibers, but this is a xylem fiber. How do you know it's a xylem fiber? Because it's in the xylem, dummies. I'm just, you're not dummies, but anyway, it's a bad joke. But we have phloem fibers too, and you'll see them. So you have phloem fibers are in the phloem, xylem fibers are in the xylem, and we have sclerenchyma fibers, which are separate. Okay, that's a concept that doesn't get clarified much. But now, this slide has a lot to do with, oh, wait a minute, I knew there was something I wanted to mention. Fibers makes me think of the dollar bill. The American dollar bill is a currency that lasts, the paper currency lasts a lot longer than some of the other countries' current currency because it's got fiber in it, flax fibers. And flax, it's actually in bond paper and cigarette papers too, but it gives a durability to the American paper currency. We don't produce it in this country, in fact, but we import it and put it into our dollar bill currency. All right, now, this slide is intended to be to tie together plant morphology and plant physiology. And we mentioned function early on, and when you say function, you need to think of chemical function, because physiology of plants is how plants function chemically, how they work. And this slide ties topics that you talk about in physiology in with morphology. And that's what's going on here. Photosynthesis, transpiration, and respiration. The transpiration, by the way, is how that water gets up to the top of the plant in the xylem. Now, as we said, or should have said, that the term, we've said it more than once, the terms morphology and anatomy are interchangeable, and they're also tied in with physiology. Now, when you think about the functions of roots, we've got anchorage, support, and storage. But the Senate here in 46, 2 years before I was born, left something out. What he left out, I'm shocked, but he left out the absorption of water and minerals. That is an important function of roots that he didn't get included. But we'll talk more about that when we get to plant physiology, Roman numeral 7. It'll be a while at this rate. But Now, before we leave this slide, the flower and the fruit are reproductive, and seed are all part of the reproductive system. And they're really compound organs because a flower is a combination of a stem and leaves. And the leaves that we'll call it when we get there are sporophylls, SPOROPHYLLS, a very complex concept to the average botanist couldn't tell you what a sporophyll is, I'm sorry to say. Today, they just can't. But now, any questions about this? As you might remember back when the first presentation, I'd made a great deal of talk about, and it had to do with the that article, The Essence of Plantness, that was in the newsletter, trying to get you to read that in the last newsletter, but talked about the surface area to volume ratio strategy of plants, and that has to do with the efficient absorption of water and minerals. Now, this slide is primarily aimed at trying to get you understand the modular concept of plants. Plants are modular. That means they're made up of repeating units. A module denotes any discrete constructional unit that is repeated as the plant develops. I'll repeat it. A module is any discrete constructional unit that's repeated as the plant develops. An example would be a flower, a bud, a node, which is a place where leaves appear, even an internode, the region between. These are repetitive units, and that's even the leaves would be examples of that, and branches, repetitive modular units. Now, growth is localized in regions called meristems. We have the apical meristem of the shoot or up here. We have the apical meristem of the root down here, covered with a root cap, which is protected as it burrows through the soil. And we said it before, and it's not, it would be good to repeat it. A meristem is a tissue that undergoes rapid mitotic cell divisions. It's generally localized in the root meristem and the apical meristem, and it can be permanent or temporary. But the point is, a meristem, think rapidly dividing cells in little brick-like shapes, because that's what they look like. And they're fundamentally young parenchyma is what they are. Notice that this blow up here, the apical meristem, it's not only that up here, the terminal, but it could be the same here in this axillary bud. At the tip of an axillary bud, if you blew it up, magnified it, it would look just like that. And this bud primordium and a leaf primordium is what you see here. Think primordium, think young. Now, the three tissue systems that come from the meristematic tissue that we've talked about, we said four tissues: meristematic, then with these other three that come from it, we have a dermal system, a ground tissue system, and a vascular system. And these are called the primary meristems that come from the apical meristem. So in the following slides, we'll concentrate on more about the protoderm, which is here, the vascular system, which is the procambium, and the everything else, the ground tissue system, when we get there. But the ground tissue system is primarily located in the cortical regions and the pith regions. Cortex is not a tissue, pith is not a tissue. The pith is the center and the cortex is the periphery. Now in the dicot root down here, or eudicot root, there is no pith because the vascular core of steel, which is on the matching, is vascular skeleton and is central. In other words, the vascular system in the root is the solid core in the center. You can't see the endodermis, even though they tried to label it, and you certainly can't see the pericycle inside of it in this, but we'll see it. In fact, the new slide I've added will be exactly this. We won't get to it today at this rate, I don't think. Okay, all right, so what's in the cortex, or what's in the pith? Parenchyma, That's right. And the cortex could have parenchyma, and it could have collenchyma or even sclerenchyma. Notice there's no, in a monocot stem, there's like corn, there's no specific cortical region and pith region that we can refer to. The bundles, the vascular bundles are scattered, whereas the vascular bundles in a dicot like sunflower are scattered, are in a ring, a concentric ring. We'll talk more about that. Now, I have props that I'll refer to from time to time, but I couldn't find my prop of the heart of a palm that shows cross section that shows that a palm stem, if you whack it, cut it off and make a section of it like this, you're just going to practically see hundreds of thousands of bundles with very little ground tissue, very little parenchyma. So corn is like this with a lot of spongy stuff in between the bundles, but in a palm, which is arborescent monocot, it's not true wood, but you see the reason it's so hard is just solid bundles. Okay, now I put that arrow in, it's not in your slide, but it's to remind me to tell you that back on that concept slide, when I brought up mesophyll before, there's two in the leaf. We'll have two forms of mesophyll, the palisade mesophyll up here and the spongy mesophyll here. Both are very photosynthetic, but you can see my criticism of the concept slide, why mesophyll would better be part of plant anatomy than morphology, because this is the only place mesophyll occurs. It doesn't occur in the stem, it doesn't occur in the root. Actually, there are some photosynthetic cells that do occur in some stems, but they don't really take the form of this mesophyll. All right, now we're primarily focusing on the big three, the protoderm, the ground meristem, and the vascular cambium. The epidermal tissue, there's an epidermis in the leaf, there's an epidermis on the primary growth of the stem, and there's an epidermis on the primary growth of this root. Now, when the stem and root get to woody stages, that epidermis is gone. Something else replaces it. Now, the ground tissue is the everything else tissue. Well, when you're not talking about epidermis and you're not talking about the vascular tissue, the veins of the leaf of the stem, or the vascular down here, it's the stele. If you're not talking about epidermis and you're not talking about conductive tissue, the only thing left is the ground tissue. That's the everything else tissue. And we have no pith in this dicot root, but there's the cortex that has primarily parenchyma. It could have collenchyma, sclerenchyma. We have a pith up here in the stem of this dicot that has parenchyma here and some cortical tissue out here. And then we have this ground tissue in the leaf. What is it? I'm going to pause and go back to this previous slide. It's the mesophyll. Okay. All right. So the ground tissue in a leaf is primarily the mesophyll, both palisade and spongy mesophyll that do photosynthesis. Questions? None. I hope that's a good thing. All right, now we have, not only do plants and animals differ at the cell level, plants differ from animals in how they grow. Plants have indeterminate growth, but you got to think about it. Flowers and leaves have determinate growth, but the whole plant, when you think of the plant as a whole, it exhibits indeterminate growth, which is pointed out in the Essence of Plantness article. That means that growth continues throughout the life of the plant. In the case of the bristlecone pine, that's been 4,800 years. The bristlecone pine, mostly, if you look at the specimen, it's dead, but there's living parts to it. In fact, any tree out there, if you look at, is more dead than alive in terms of tissue. But it's in trees, the dead tissue functions and support quite a bit. All right, now before we go further, we're going to concentrate on talking about behind the root cap. There's a region of the meristem where cells are dividing. Then there's an elongation region, which you'll emphasize in the next slide or so. Then there's a differentiation region. So here behind the root cap, we have a meristem, elongation, and differentiation. And the differentiation is easy to pick out because you start seeing root hairs. And then if you look inside, you'll start seeing some procambium developing, but it's not showing here. These three regions, the meristem, the elongation, and differentiation regions, are easier to point out in a root apex than they are a stem apex. But this stem apex, of which we'll focus on separately in a moment, you can see the procambium developing already. The veins in the bundles, vascular bundle of a stem will turn into a vein in the leaf. And then the meristem is where the rapidly dividing parenchyma cells are. These are storage parenchyma in the center, will be part of the pith. And then we have a leaf primordium is a young leaf, and a bud primordium is a young bud. That little area right there, if we enlarge it, it would look just like that. Okay. Now, when you think of growth, you think of the increase in biomass, and it comes from two things, two processes in plants. It comes from cell division, and it comes from what you see here, cell elongation. So growth is a function, in short, growth is a function of cell division, but what we see here is cell elongation, not division. These are corn seedlings that have been marked, and after 36 hours, we can see that how much it has increased in size, but that increase in size is primarily due to the elongation of the parenchyma. Instead of being isodiametric, they're now rectangular and quite long. And the three regions I've already pointed out are the meristematic zone and the region of elongation differentiation. The two that I've highlighted are where the growth takes place. Division in the meristematic zone, elongation in the region of elongation. And again, if you want to figure out where you are, you start seeing these little projections on the outside. These are root hairs, and that tells you that you're getting to the region of differentiation. And in a moment, we'll be able to maybe see some of the internal differentiation like procambium. And in the case in the stem, you can see this here. You have the three primary meristems in the stem, coming off of the apical meristem are the procambium. That's the beginning of this vascular bundle right here, procambium. And then the protoderm, which is out here, the epidermis to be, and then the everything else, the ground meristem, which could be cortical and could be in here too, in the pith region. And again, the bud primordium and a leaf primordium. Any questions? This is on a bean plant. Now, this is a little confusing to some people because of this color, I guess. I'll get to that. And this, the black that you're dealing with here is the newest tissue. Now, this has to do with why you have to mow your lawn if you have a lawn. This is why you have to mow it, because and continue to mow it throughout the season. Because unlike dicots, you cut off the apex, you cut off the terminal growth. In the case of a monocot, you cut off the apex, you have it, because there is a basal or intercalary meristem. Basal referring to the base of the node, okay? At the base of the internode. The internode is a place between, here's a node, and here's the internode. So right here is a region, a meristematic region of growth. Here's one, here's one, here's one. And so cut off the top up there, it doesn't matter. This monocot is gonna increase in length at this intercalary or basal meristem that's situated at the base of the internode. And the confusing thing to me is that the black is the newest, But if you happen to go out, and I've looked, I've seen it, but I've never photographed it. And I'm sorry I haven't, because on the internet, you can't find what I'm about to point out. When you burn wiregrass, you'll see the black growth will be the old black growth will be at the tip, the opposite of what's depicted here. And you'll see the new green growth down below. But it's just the total opposite from this concept. But in real life, it shows up. They shouldn't have, the artist that I took this from, I wished he hadn't have made the newest stuff black, because in wiregrass, it's, when you see it's been burned, it's exactly the opposite. Now, here's, we're making progress here, because at this point, we're transitioning between primary growth and secondary growth. And I put a date on that down here because I modified it, and this is how I modified it. The artist should have put a line between these vascular bundles, because the vascular cambium that we're going to soon to be talking about, that comes from the procambium. This vascular cambium tissue comes from the procambium. What comes from the procambium? Xylem, that's right, phloem, and ultimately the vascular cambium that's going to form the secondary tissues, one of the secondary tissues. That's what's going to form the other one, the cork cambium. But in this diagram, everything is continuous. In this diagram, it's not. But if you have a one layer in here that's called the vascular cambium that's not really well seen at this point. The cambium that's between the bundles is called interfascicular. All right, let's break the word down. Fascicle. A fascicle is a bundle. An inter, like an interstate, connects states. So an interfascicular bundle is an interfascicular vascular cambium is a layer that connects vascular bundles. It's not shown here, but that's the heart to how, that's the key to how we go from primary to secondary tissues. Now, this is a cross section of a sunflower dicot. I believe it's a sunflower. I can't see up here, but anyway, it's a dicot. And I think it's actually Helianthus, but, and this is covered in the top, so I can't really see it. But all right, so we've got it. Now, is sunflower a woody plant? No, but it's a herbaceous plant that gets a little bit of woody tissue if you leave it long enough. And so we've got vascular bundles in a concentric circle and a pith region and a skimpy little cortex region here. And these are fibers. And since they're in the phloem area, guess what we call them? Phloem fibers. They're not sclerenchyma. Now, there's one layer here, and one layer only is called the vascular cambium. The vascular cambium that differentiates on the outside is called phloem parenchyma. The vascular cambium differentiates on the inside is here in the bundle is called Xylem parenchyma. And so eventually we get some going here too, and this is the interfascicular vascular cambium that I was trying to say should have been labeled here. This is exactly the same thing. So we get a continuous vascular cambium ring in a concentric circle. And by the way, this is very much has to do with that other diagram I sent you last night, where we have periclinal divisions going on this way, and you have anticlinal divisions responsible for increases in girth. Anticlinal divisions are radial cuts. And so that's how the ring stays continuous, by anticlinal divisions, where periclinal divisions is the way you form phloem parenchyma and xylem parenchyma. Okay, that's pretty heavy stuff. Any questions? It kind of bothers me nobody's asking any questions. I hope you're still there and thinking. I hope I haven't lost you. But, okay, now, all right, is this possible? No, it's not possible. Alan Heilman was my teacher at Tennessee, one of them, and this is from his lab manual. He gave me permission to use it. About late in the 70s is when I was there. And this is not possible because the elongation of any woody plant takes place in the up here in the apical meristem and down here in the roots meristems. And so this is not possible. The tree, a branch that will be in the same general location in 1920 as if the branch is still there, and 51 years later, it should be about the same. Now, this is comparing, we say, gee, aren't you talking, where are you talking about secondary tissues? Why are you still talking about primary? Well, I'm sure there's a few more points we need to make. I already said the vascular bundles are arranged in a concentric circle, and we have the intervascular cambium and the vascular cambium in a dicot. The monocot doesn't have a real pith and cortex because the bundles are scattered. All right, now what I'm getting at is a concept we're about to bring up is the difference in a closed bundle here and an open bundle here. All right, this slide and this one bring out that point. All right, the monocots have closed vascular bundles because they're closed to further development. The dicots have open vascular bundles because they're open to further development. All right, now here's the phloem fibers I talked about. Here is the vascular cambium, and here is the xylem tissue. It's out here. So this is a sectional view of right here. This is the outside, this is the inside. Outside, inside. All right, now the next slide, we're going to see a real view of these closer up pretty soon, okay? This is the herbaceous dicot like sunflower. This is a section of corn showing a close-up bundle. In fact, this is the same as this. Okay, now, all right, this is an airspace, by the way, that has developed. There's nothing there but it's hollow. And this vascular bundle is closed because it has no vascular cambium in it. There's no vascular cambium in it. This is a vessel element. These are phloem companion cells and sieve tubes. In fact, let's do that. Let's go back. I want you to go back and think 3D. Let's go back to, hang on. Go back to right here. All right, now, remember the sieve tube and companion cell and cross section? Think this view right here, because that's where we're going. That's what we're doing right now. Which one's bigger? The sieve tube element is bigger than the companion cell. Now let's go back. Navigation here is kind of like I feel like I'm going through molasses. All right. Okay. All right, now. All right, so now the small diameter cell is what? Companion cell. The larger diameter is the sieve tube element. Now, if you look carefully, you can see a plate right here. It just didn't show up very well, but the sieve plate is only going to show up once in a while when you happen to cut through that part where the two sieve tube elements meet. And so that's not going to be every cell you see. I don't have a good slide of that right here in front of me to show you, but anyway, this is the phloem, primary phloem. This is the primary xylem. And actually that's a trachid, and these are vessel elements. How do I know that? Because that comes with experience. But anyway, there's some phloem fibers out here too, and out here in this bundle. And again, in palm, this is solid bundles practically, unlike corn. All right, what's wrong here? This is not properly labeled. The vascular bundle, I took this from a book and it just was poorly done. And somehow or another, the marker got moved, but the bundle is here, the vascular bundle. And one other thing needs to be pointed out is, and I can't see it because let's see if I can. Okay, closed bundles have no what? Vascular cambium. That's what that's saying. All right. Now it's 11. We're going to go back a few more minutes, maybe 5, no more than 10 for sure, and we'll be done, which is better than the hour and a half that we usually go. But I got about where I wanted to get in the secondary growth region. And I'm going to introduce a few things before we cut loose. What is wood? Wood is secondary xylem. And by the way, when you see a 1 and a degree sign, it's a symbol for primary, like the handout I sent you last night. And the counterpart is 2 with a degree is a secondary xylem. Secondary anything is the symbolism we're trying to indicate here. But wood is primarily made of secondary xylem. The inner bark is made up of secondary phloem. The outer bark comes from cork cambium and includes cork tissue. Now, cork is another name for cork is PHELLEM, the phellem. And there is another tissue that the cork cambium develops called phyloderm that we'll get to this time or next time. But all right, now when you girdle a tree, you're taking the bark off, which means you're taking the inner bark, the phloem, and the outer bark out here, and you're girdling a tree. And that's a slow way to kill it, but that's the way that trees are mechanically girdled. Now, this is one of my favorite slides from a very old St. Regis paper company chart. It was about 3 foot by 3 foot, and I photographed it and have used it because it shows a lot of good things. It shows the heartwood and sapwood. The heartwood is functional, but it's functional in support because it is not functional in conduction anymore because it's clogged up. That's why it's dark. The sapwood is functional in support and conduction. And you can see the growth rings here. We'll talk about, well, actually here, Springwood and Summerwood. Springwood is rapid growth and Summerwood is slow growth when the water is not as abundant. And in the true tropics, true tropical rainforest, you don't have annual rings. You have diffuse growth, not like you see here. So drought areas in the tropics can mimic a spring and summer wood, but this is the growth rings that you can count on a tree. Why is the heartwood clogged up? Because plants don't have an excretory system, so their garbage gets locked into their cells. Tannins, that's what are bitter, alkaloids and other compounds build up in plants. And the heartwood is where they get retained, and it's a structural support function. But the conduction is here, in the water and minerals is here. Notice this vascular cambium is one layer. It forms secondary xylem here, it forms secondary phloem here. And this is the secondary phloem, which is commonly called the inner bark. And this outer bark is a function of the cork cambium that we'll talk more about next time. Let's see. Yeah. All right, I think we'll do this slide and quit. Yeah. All right. The pith is what's going on here. The pith gets obliterated. What do you mean? Well, when you keep producing wood in the stem, I'm talking stem, this is basswood, when you keep producing wood going toward the center, guess what? The pith in the center gets crushed, obliterated. And eventually the stem loses its pith and becomes solid vasculature after years of pushing to the center. This gets obliterated. The parenchyma and the pith gets obliterated. All right, we just finished talking about primary growth, which is the growth from apical meristems, but now we're talking lateral growth, and that has to do with the wood and the bark. What is this? Oops, this is a vascular ray. Vascular rays are made up of a xylem ray and a phloem ray. The phloem ray gets dilated because these are not real rigid out here except for a few fibers, and you get phloem parenchyma that easily thin-walled that dilates because it's like a racetrack. If you're on the outside, you're having to run further to keep up. So in here, when the girth increases, it's not as much of a problem. Plus, these cells are rigid-walled xylem cells. What does a vascular ray do? It's responsible for horizontal translocation. And if you want to cut a piece of wood and let it dry out and there's cracks, guess where the cracks are? The cracks are right here where this is xylem parenchyma. So there's this is mostly xylem fibers, xylem tracheids, and vessel elements, the larger diameter vessel elements. But this area here and here and here is xylem parenchyma. It's where the cracks in wood develop. Well, you can count the year's growth. This is Summerwood's, Summerwood, Springwood, Summerwood, Springwood, Summerwood. 3 years of growth and the vascular cambium is here. All right. All right, we're going to pull the plug there, and it didn't do too bad. Let me come out of here and see what, if everybody's still there. Let me stop sharing. Okay. Everybody seems to be still here. That's good. Okay, in the chat, somebody said something here. Let's see. Are individual xylem fibers one cell thick? All right. Yes. Yes. A fiber is only one cell thick. Yes, that's true. All right. Any questions? Okay. This is more like a normal Hadaway today. So, all right. Bye-bye, whatever that is. All right.